All right, good evening, everybody. We are so excited for our summer 2022 research experience students to present this evening. My name is Jen. I am the program and outreach manager for Headwaters Science Institute, as well as a mentor for the program. I have over 12 years experience of teaching science research, as well as marine biology, AP Environmental Science, AP Research, as well as working in the field in the marine sciences. So very quickly, I'm going to bring in one of our other mentors, DJ, to go ahead and introduce himself a little bit, because you'll see him coming in and out as he introduces his students. So thank you, DJ, so much for joining us today and the work that you've done with our students over the course of the summer.
Yeah, well, hello everyone. As she mentioned, I work for Headwaters as well. I typically work as a data analyst and mentor, and I'm also an adjunct faculty at one of the community colleges here in Texas. I specialize in a wide variety of research, including phylogenetics, ecology, evolution, and I've taught various types of courses as well, like microbiology, anatomy, and physiology, and whatnot. But I really specialize in sort of mentoring all of the students within the Headwaters programs and helping them create, really fascinating and unique projects that sort of fit their curiosity. So it's a pleasure to be here.
Thank you so much, DJ. And also this summer, we have Christina Labuglio, who also was acting as one of our mentors. So thank you, Christina, so much for the time that you spent this summer. If you can go ahead and introduce yourself a little bit to everyone.
Yeah, hi everyone. It's great to be here. Like Jen said, my name's Christina. I'm a research tech for Auburn University. I work at their shellfish lab. So right now I head oyster reef restoration projects and take part in oyster aquaculture and farming of oysters for research purposes and commercial purposes. But I've also had my hands in some shark research with OSearch, and that's where I met Jen about 8 years ago. And my master's thesis in Antarctica, looking at glacial ice melt in Western Antarctica. So it was a pleasure to have all my students, and I'm really excited for everyone to see what they've done this summer.
Awesome. So to kick things off, we actually have one of Christina's students. So we're going to go ahead and introduce Ian. So Christina, if you would like to tell everybody a little bit about Ian.
Yeah, so Ian, you're the first one. You're setting the level for everyone. So up first is Ian. He's from the South Bay, San Jose area in California. And he's a senior this year, so that's exciting. College is coming up soon, and he is striving to go into environmental engineering, which is really fun. He's done a lot of hard work this summer. I'm super proud of him, and it's a really cool project. So I can't wait for him to share with you guys his project on eutrophication in California. So Ian, take it away.
All right, hello everyone. This is my presentation on understanding water quality in urban versus agricultural areas. So to understand water quality, we need to understand the types of pollution that are involved in water quality. So the first type is point source pollution. This would include anything that is directed from pipes into water, and this usually occurs when there's a lot of rainfall because the excess water is discharged in the streams. And overall, we haven't seen this be a huge problem recently because of some legislation that has been passed. And that leaves the elephant in the room, which is non-point source pollution. Non-point source pollution would basically include anything else that doesn't directly run into the water through a pipe. So that can include pollution from farms, pollution from the city, pollution from natural areas. And then one of the main types of non-point source pollution is runoff. So for this study, we'll be studying the difference and effect of urban runoff and agricultural runoff, and their definitions are pretty intuitive. But now for my first site, This site was important for studying agricultural runoff, Ogier Ponds, because it is located near some agricultural areas. At one point, there was a winery nearby and also a hog and tomato farm. And currently, there is a cherry farm right next to it. So I studied this site in order to understand the effects of agricultural runoff. And I studied these next two sites, Almaden Lake and Hellyer Lake, which are urban regions, to understand the effects of urban runoff. Interestingly enough, Hellyer Lake is connected to Ogier Ponds. It's just a little bit farther north, so it's more likely to be affected by urban runoff. And also, Almaden Lake is affected by urban runoff. And right now, they're undergoing a project to repair or improve the water quality by sending down this stream, this restored creek right here, because the water quality in Almaden Lake is known to have a higher concentration of mercury and a higher temperature. So now, to understand the effects of urban runoff and agriculture runoff, one of the main areas where we can look is eutrophication. Eutrophication means man-made algal bloom and is usually the result of excess fertilizer. The reason that eutrophication is not good is because it can use up all the oxygen in the stream, because when bacteria consume algae, then they deplete all the oxygen, and oxygen is essential for aquatic life. It's also essential for the health of streams and many other areas. So with this background, it leads me to my research question, which is water quality at Ogier Ponds, which is an agricultural region, compared to Almaden Lake and Hellyer Lake, which are urban regions. The independent variable that I used was time and location, and the dependent variables were pH, iron, copper, nitrate, nitrite, hardness, mercury, and total alkalinity. My hypothesis for this study was that overall, Ogier Ponds will have a poor water quality because of fertilizer from agricultural runoff. So some of the materials that I used was a verified 16-parameter water quality test kit, which was used to observe pH, iron, copper, nitrate, nitrite, and total alkalinity at each of the locations. So in my first result, I tested for pH, and I found some significant difference between the agricultural region and the urban region. And this is an indication also that there is significant difference. And also, however, I did not find any significant change over time in the amount of pH. There was insignificant difference between the amount of iron in the creek, and there was also no significant change over the time. This was also the same for the amount of copper, the amount of nitrate, the amount of nitrite. However, the hardness level showed some difference with a p-value of 0.03, indicating that there was some difference between agricultural regions and the urban region. There was also, however, no drastic shift over time in the amount of hardness. And finally, or not finally, but also there was no mercury detected in any of the water, and I will get to this in a moment in the discussion. In Hart, for total alkalinity, there was some difference recorded between the three sites, and there was also some change that was seen over time that was, compared to the other site, was a little bit more drastic. So some of the reasons for understanding the significant results, at least for pH, is the presence of fertilizer in agricultural runoff. Fertilizer tends to have a pH between 6.5 and 7, and this is important to know because this can often lower the overall pH of the water. Another major factor is the total amount of alkalinity. Alkalinity would include like the bases in the water, which includes bicarbonate, carbonate, hydroxide. And this total alkalinity is positively correlated with pH. So when there's a lower amount of it, there's also generally a lower amount of pH. And also the significant difference between alkalinity can be attributed to the sediments in the water when water goes through and pulls off some of the parts from rocks, as well as hardness can also be contributed, the difference, to the types of sediments that are at the bottom of the creeks. There was also some significant change over time in the total alkalinity, and this can be attributed to the pre-rain season because generally when it's not raining, water flows more slowly through these creek areas and is able to pick up more of the minerals that are in these rocks than it would be able to during a rainy season when the rain is pouring. There were also some results that were not that as significant, like the difference between iron and copper nitrite, which did not vary significantly between the study locations. A reason for this is that generally during drought season, water quality is cleaner because of less rainfall that carries in less runoff. Also, only alkalinity underwent dramatic change over time. Also, remember that mercury was not detected in any of the water. This is not to say that there was no mercury, but generally mercury stays at the bottom of the water in the rocks. So it is only detected if you mix up the rocks in the water. If you stir it up a little bit, then you will detect certain amounts of mercury. Some important future research questions that I can do is, how does weather pattern over a long period of time affect water quality based on the area? Also, how does the water quality affect the associated biodiversity in water bodies? And finally, what are the economic implications for poorer water quality? As I wrap up this presentation, I would like to give a few shout outs to some important people. First, my parents for enrolling me in Headwaters and letting me use the car to get around to different places. That was very useful for me because I wouldn't have been able to collect the samples without them. Also, thank you to Headwaters for overseeing my research project. Thank you to Christina for walking me through the whole process and making it something that seemed really daunting a lot more manageable. And then finally, thank you to my dad and Sam for collecting data while I was in Los Angeles and Seattle. That was a big assist to me. And also, it's not on the slide, but I'd also like to say thank you to my peers for peer reviewing my paper and getting through this process with me. It's been awesome, you guys.
All right, amazing job, Ian. So right now what we're gonna do is pull the mentors in just in case anybody has any questions. And Christina, if you have anything to say, you did such a great job. So proud of him.
Yeah, Ian, I just want to start with it was a pleasure working with you, and your whole project turned into something. I'm glad you learned something. I'm glad I made it seem a little less daunting for you, because you did an excellent job with collecting the data and working through everything and making sure you're getting the research done and your manuscript done. So just great job for me. I've asked you plenty of questions. So if anybody else has questions.
Jay, do you have any questions for Ian?
Yeah, I'm curious about, how was your experience doing field work? That's one thing that some people may not enjoy doing. But overall, what was your experience with conducting that kind of, data collection?
I enjoyed it quite a bit because I'm a big outdoors person, so it gave me an excuse to go for a little walk every day to the creek and collect my data, and also give me time, like when I was walking back and forth between sites, to think about like, what does this data mean, or like, I don't know. It definitely depends. If you're an outdoor person, you like it more. If not, maybe it's not your thing. But I'm an outdoor person, so it was great for me.
Speaking like a true field ecologist or a field biologist.
Yeah. So being that it's so close to your house, like, are there any changes that you think that you might do in your own personal household, given the information that you found out from researching the creek?
Maybe I start to test the water in my house just to see how what kinds of filtration it goes through, because I know at least from Almaden Lake that water ultimately comes to my house because they have a reservoir right after it where they clean up the water. Valley Water, that's the local water company. So it'd be interesting to see what stays in the water and what doesn't stay. And also just I don't know, I'd like to do some research on maybe how much it cost to clean the water, depending on what types of contaminants are in there. But yeah, it's definitely sparked a lot of interest for me.
Awesome. Well, great job. We're all so proud of you, and we're excited to publish your paper very, very soon and see where you go from here. So congratulations.
Yeah.
Thank you.
Good job, Ian.
Right.
Great job.
Yes, great job. All right. So I'm going to go ahead and leave DJ in because the next student we have up is Shreef Shritha, can you say her name for me properly?
Yeah. So I'd love to introduce Srinita from the Bay Area. Just before going into sort of her work, some of her hobbies include art, dance, and playing the violin. I don't know how she has time to do any of those things when she does such amazing research. Truly throughout this summer, I've had the privilege of being her mentor, and you're going to learn about the fascinating and very advanced research that she's conducted of studying the evolution of crystalline genes throughout vertebrates. So I mean, yeah, you can take it away, and I hope everyone is amazed at your work as I am.
Thank you. Hi, my name is Shanita, and today I'll be presenting my research project, which is a phylogenetic analysis of the evolution of the CRYAA gene across vertebrates. First of all, what is a phylogenetic analysis? Phylogenetics is the study of evolutionary relationships among or within groups of organisms. In many ways, it is the foundation for comparative biology and the study of life systems. The goal of a phylogenetic analysis is to construct a phylogenetic tree, or phylogeny, which will give you information about how closely related these groups of organisms are to each other. It also allows you to visualize the history of descent by showing you which species had common ancestors and how they diverged from each other over time. Now, what is the CRYAA gene, and why is its evolution a topic of interest? The CRYAA gene controls the expression of alpha-A crystalline, which is a soluble lens protein occupying 40% of the proteins in the vertebrate eye. Its function is extremely important to vertebrate vision because it is responsible for maintaining the transparency of the lens and its refractive index, which is the ability of the lens to bend light. In addition, it is a small heat shock protein, meaning that it prevents the misfolding and denaturation of proteins. Alpha-A crystalline is believed to be extremely conserved, meaning that it hasn't evolved over time. Here are pictures of the protein in humans, sheep, and mice. Although the evolution of alpha-A crystalline has been studied, there hasn't been a study on how the CRYAA gene that controls it has evolved across vertebrates, which is why I decided to construct a phylogenetic tree of this gene. If it is able to accurately indicate the evolutionary relationships between different organisms, it can be used to study vertebrate evolution, and this could benefit the field of phylogenetics. My research question was, how has the CRYAA gene, critical to vertebrate vision and the expression of alpha-A crystalline, evolved across vertebrate species? The independent variable was the individual nucleotide and amino acid sequences that were inputted into the analysis. The dependent variable was the evolutionary relationships determined by the phylogeny. In this project, I tested the null hypothesis that there is no sequence differentiation in the CRYAA gene across vertebrates. Since no studies on this topic have been done, there is no evidence in support or against this hypothesis, so I assumed the null. My methods consisted of two parts: a phylogenetic analysis of the CRYAA gene and a phylogenetic analysis of the alpha-A crystalline protein. For the CRYAA phylogeny, I started by retrieving nucleotide sequences from the online repository GenBank. I chose 31 total sequences, which were taken from 20 different species and 15 genera. The first step was to prepare the sequences for the analysis. I imported them into Mega 11, which is an application that I use to align and trim the sequences. I then constructed a phylogenetic tree using the corresponding function in Mega. For the alpha-A crystalline phylogeny, I repeated the same process with one added step in sequence preparation. To obtain the amino acid sequences that I needed, I first translated the nucleotide sequences from the CRYAA gene into amino acid sequences using a tool called XBC. In XBC, when you input a sequence that you want to translate, it gives you 3 different reading frames, and only one of them contains the right amino acid sequence. To determine the correct one, I looked for the frame containing the longest consecutive red highlight. So for example, in this picture, the first frame would be the correct one. For instances where it wasn't as easy to tell which one was correct, I looked for frames containing amino acids that are characteristic to the alpha-A crystalline protein, like SRLF. After aligning and trimming the sequences in Mega, a phylogenetic tree was constructed the same way as the first one, By the end of this process, there were 4 total phylogenetic trees, 1 original tree and 1 bootstrap consensus tree for both the gene and the protein. This brings us to the results. Here's the original tree for the CRYAA gene. It is based off of the original data that was inputted into the analysis. The lengths of the branches correspond to the evolutionary distances between each organism, so longer branches indicate greater distances. The number 0.2 at the bottom is a scale. The bar above it would be the length of a branch for an evolutionary distance of 0.2. The numbers on the left side of the tree are called bootstrap values. They denote the confidence out of 1 that each clade is where it's supposed to be. A clade is a group of organisms that share a common ancestor. So for example, this would be a clade. Overall, we can see that the tree did a pretty good job of distinguishing between different clades, as it does show distance between them. The tree is color-coded based on genus, which makes it easier to observe how species of the same genus all clustered together. Moving on, here is the bootstrap consensus tree for the CRYAA gene. It is based off of resampled or bootstrap data rather than the original data. One thing to note is that the purpose of this tree is to depict the relationships between each clade, but the branch lengths do not mean anything and they are not indicative of evolutionary distance. The bootstrap values are much higher on this tree, meaning that the confidence that the position of each clade is accurate is much greater. Here's the original tree for the alpha-A crystalline protein. Right off the bat, we can see that there isn't much evolutionary distance between any of the species. The uppermost branch doesn't have 2 lines extending outwards from it, and this indicates that it wasn't able to detect any sequence differentiation in the species that it encompasses. So clearly, this tree isn't very informative of the evolutionary relationships between each organism. And in addition, the bootstrap values are extremely low. And lastly, here is the bootstrap consensus tree for the alpha-A crystalline protein. Although the bootstrap values may be higher on this tree, it doesn't really tell us much about vertebrate vision, and it's not a very informative tree like the CRYAA gene trees were. The null hypothesis that the CRYAA gene has not evolved across vertebrates was unsupported by the results. The CRYAA gene trees proved that it has evolved over time, as they were able to distinguish between different vertebrates based on their sequences. Overall, the tree does appear to be pretty accurate. For example, we can see that all these rodents clustered together, the mouse, the rat, and the guinea pig. We also see that all of these bird genera were put together, and the sheep and the cattle were put in the same clade. So the tree does appear to be pretty consistent with the accurate evolutionary relationships between these different species. Although the CRYAA gene may be a good indicator of the evolutionary relationships between different genera, it may not be as useful for distinguishing between species within those genera. For example, if you pick any of the bird genre at the bottom and you look at the species within those genera, you can see that it doesn't show much distance and the branch lengths are not significant enough to be noticeable. The alpha-A crystalline protein, on the other hand, is not a good indicator of evolution, as the trees weren't able to distinguish between different species and were supported by very low bootstrap values. In conclusion, the CRYAA gene has evolved and could be a useful indicator of the evolutionary relationships between different genera, while the alpha-A crystalline protein is too conserved to be used to study vertebrate evolution. For possible extensions to this project, it would be interesting to construct a phylogenetic tree of the CO1 gene, which is commonly used for DNA barcoding in phylogenetic studies. With the CO1 tree, it would be easy to establish how accurate the CRYAA phylogeny was at determining these relationships by comparing the 2 trees. For other research, studying the evolution of genes controlling other heat shock proteins could yield pretty fascinating results like this one did. And this could also lead to the discovery of other genes that could be used for future phylogenetic studies. And lastly, instead of studying the evolution of genes, studying the evolution of alpha-A crystalline in relation to other lens proteins and how they evolve from other proteins could be a pretty interesting project as well. Thank you so much for listening. And I just wanted to say thank you so much to my mentor, Mr. Daniel Dudek, for all the time that he spent guiding me through this entire process and helping me design this project. Without all of his time and his constant support, I wouldn't have even thought of doing a project like this. So thank you so much. I very much appreciate all the time that you put into this. And thank you to all my peers who reviewed my manuscript and also Headwaters for helping me through this process. And here's my works cited. Thank you.
I mean, wow. You pull Christina into, amazing job.
Thank you.
Yeah, great job. Again, like I mentioned before you started, I was very impressed with the amount of work that you were able to accomplish. And I hope everyone can keep in mind, whenever she signed up for this, her current grade level is 9th grade, right? Right? Or are you going into 10th grade? Going into 10th grade. So, this level of research, this is almost like master's level work that she's doing, pulling gen bank sequences. I mean, it's truly amazing what she was able to accomplish in such a short period of time. So yeah, great job.
Thank you so much.
Yes, job well done. Christina, do you have any questions?
I'm trying to think of one, and
I have a very basic question. Like, so the methodology that you use, could this be applied to additional? And you may have already addressed it, but I was just sitting there watching you like additional gene sequences to look at other avenues as far as evolution and relationships amongst organisms.
Yeah, definitely. Mega is a really useful software. It gives you all of the functions that you need to align, trim the sequences, and also construct the tree. So yeah, it could be applicable to pretty much any gene sequence that you can find online.
So do you think that it might be helpful for maybe healthcare when looking at inheritable diseases and things like that?
Yeah, definitely. Also to study like the mutational rates of different genes and yeah, I definitely think it could be useful for healthcare because genetics is a really important part of healthcare.
It's always changing too, so it's always great to have a routine and a process that's something you can keep up with. So that's amazing. Yeah. Anybody else have any questions?
I don't have a question, but I just want to say that was an amazing project. And I myself learned a bunch. I don't know much about amino acids and proteins and such. So I think it was a really good project, and I enjoyed listening to it.
Thank you so much.
Well, great job. So I'm going to pull Christina. So up next we have Tiffany. Here she is. So I'm going to go ahead and let DJ introduce Tiffany and go ahead and pull in her presentation. So welcome.
Yeah, so again, another stellar student that I had the pleasure of mentoring throughout this process. Tiffany, I think you're going into 11th grade. And, some of her hobbies include baking and watching movies. So I'm sure when she wasn't, studying, she was enjoying, her Friday nights or whatever, she was doing there. And she's also from California as well. And she's going to be, talking about her research really involved sort of diets and different preferences and macronutrients and the amount of fat percentage across, the entire world. So, I'm really excited for everyone to hear, her study on this exciting topic and the extremely high quality data sets she was able to generate to address this question.
Yeah, thank you. Hi, everyone. My name is Tiffany O, and my project is going to be about the global dietary habits in relation to adult obesity. And once again, my mentor, Daniel Dudek, was able to help me with this project. So first, globally, obesity is on the rise. In 2016, 13% of adults were obese, and this was a number that tripled over the past 4 decades. And obesity varies around the world. For example, in 2016, the US had an obesity rate of 36.2%, while Japan had an obesity rate of 4.3%. But how is this obesity measured? Usually the body mass index scale, or the BMI scale, is used to measure obesity. So this scale takes into considerations one's height and weight and gives a value. And the value of 30 to 39.9 is a value that measures obesity. But what causes this difference globally? So first, the overconsumption of sugar could lead to obesity. The recommended sugar intake for a female is about 24 grams, which is about 100 calories. And for a male, it's about 36 grams, which is about 150 calories. However, many individuals often go over this recommended sugar intake, which can overall lead to weight gain and eventually obesity. Also, a healthy macronutritional intake is key for maintaining a healthy weight level. So this means a healthy balance of fats, carbs, and proteins. However, some countries have less balanced diets than others. For example, on the map on the lower right corner, we see the overall dietary patterns of each country. And according to the color difference, we can see that some countries are a bit better at maintaining healthy dietary levels than others. Lastly, an overconsumption of calories can cause weight gain. So going into the research question, how do dietary habits of each country relate to their relative obesity rates over time? So the independent variables were sugar consumption per capita, carbohydrate, protein, and fat percentage, as well as caloric intake. And the dependent variable was adult obesity percentage. And that leads us to my hypothesis. A high sugar consumption, an unbalanced macronutrient intake, a high caloric intake leads to a higher adult obesity percentage. So for the materials, most of the materials are found online. So this included online research articles and blogs to find the data, and also a data tracking table, which in this case was Google Sheets, as well as data classroom to make the graphs. So first, for the methods, the countries to be focused on were chosen to get a good global range, and the years for each dataset were also chosen. After that, it was just constant research and finding data through sources such as HELG Library, Global Nutrition Profiles, ChartSpin, and FAO Stat. Now, going to the results, for the sugar consumption per capita, we saw a positive relationship. So as a country consumed more sugar, the adult obesity rate also went up. And for this, we see the R-squared value 0.429, which represents the amount of variability in the y-axis explained by the x-axis, and the p-value less than 0.01. So a p-value of anything less than 0.05 shows that our data is significant and shows a statistically significant correlation. Now for the second data set, we saw that as the carbohydrate intake went up, the adult obesity actually went down. So this was a negative relationship. And for this data set, we saw the R-squared value 0.442 and the p-value of less than 0.01. Now going into our second macronutrient, proteins, we saw that as a country consumed more protein daily, the adult obesity percentage went up, showing a positive relationship. And we saw the R-squared value of 0.353 and a p-value of 0.01 for this data set. Now for the last macronutrient, fat, we saw that as a country's fat intake per day was higher, the adult obesity percentage was also higher. Now for this data set, the R-squared value was 0.428 and the p-value was once again less than 0.01. Now going into the last data set, caloric intake per day, we saw that as the amount of calories a country consumed per day increased, the adult obesity percentage also increased. So for this, the R-squared value was 0.41, while the p-value was once again less than 0.01. Now going into the discussion, first we saw that greater the sugar intake, greater the obesity. And a possible explanation for this could be because sugar actually slows down one's metabolism. Metabolism is a body's way of generating energy by using the nutrients an individual consumes. However, sugars such as fructose cannot be metabolized, so the body could not possibly take in the sugar one consumes and release it as energy. Therefore, it could lead to weight gain. Next, we saw that greater the carbohydrate intake, the lower the obesity. And this could be because carbs actually speed up the metabolism process and release fatty acids, which encourages fat burning. So this could actually even cause weight loss if one maintains a healthy diet with a good amount of carbohydrates. And carbohydrates actually offer greater satisfaction. So an individual doesn't have to turn to other sources to get the nutrients or satisfaction they desire. Also, we saw that greater the protein intake, greater the obesity. And this could be because excess protein is actually stored as a fat in the human body. So constantly eating too much protein could cause weight gain. Although protein is a good source for muscle repairment and organ protection, too much could lead to obesity. We saw that greater the fat intake, greater the obesity. And this could be because fats are actually a very calorie dense substance. So eating too much of it could pile up and cause an individual to gain fat. And fats are about 9 calories per gram. So once again, a constant overconsumption of this would eventually lead to obesity. Lastly, we saw that greater the caloric intake, greater the obesity. And this could be because an overconsumption of 3,500 calories is equivalent to about 1 pound of fat gain. So once again, constantly overconsuming this over a period of time could lead to weight gain in the country. So for future research, we can consider food accessibility in certain countries. For example, a certain country may have more access to protein than others, which could explain why the individuals and citizens of that country is getting more protein than other countries. Also, we can start to study additional health concerns arising from dietary choices. For example, diabetes arising from too much of sugar, and heart issues arising from an overconsumption of fat. Lastly, physical activity in relation to obesity rates could be studied. So we can examine an individual's activity and exercise in relation to their obesity rates and change in weight over time. So lastly, I would like to make some acknowledgements. First, I would like to thank my mentor, DJ, for meeting with me every week one-on-one and really giving such personal feedback to help me improve my research project and just guide me through the process. And also, I would like to thank my peers for giving me revision advice and helping me really perfect my manuscript. Also, I would like to thank Jennifer Cotton for holding meetings every Tuesday and giving us really the core information to help us carry out our research project. And I would like to thank Headwaters Science Institute for creating such an awesome opportunity for me to pursue my research in a topic I'm passionate about. And lastly, these are my works cited that helped me complete my research project and collect my data. Thank you.
Yeah, great job. Great job.
Yes.
Loved it. And I'm sure other people may have been shocked with some of her results, especially the ones where she found that, the percentage of carbs that a country consumes, actually they have a lower obesity rate, which, now in today's society, carbs are sort of demonized, right? I mean, everyone, wants to stay away from carbs and just consume, fats and proteins. But yeah, it's some really interesting, head scratching results that she found here.
I'm curious if you did any research into the way that those carbohydrates are kind of processed within the country and the results that you saw with obesity levels, because I know that we process things differently than, say, like Italy, who eats tons and tons of pastas and, different types of carbohydrates. So I'm wondering if you looked at any of the processing in factories and things like that, of how that may have influenced your results.
I just focused on the main nutrients and how people consume it. But my kind of, consensus on that was when people lean towards other things instead of carbs, they aren't getting the energy they need and they aren't feeling satisfied. So they constantly lead to other fatty substances or substances full of protein. So that's what actually causes the weight gain. So if everyone just gets the amount of carbohydrates they need, no matter what type of carbohydrate it is, it'll provide them with sufficient energy and sufficient satisfaction throughout the day.
So it goes back to having that balanced diet that we since we were little kids, right?
Yeah.
I have a quick question. Were you able, I know you didn't show it in your results, but just from you personally looking at your results and when you were collecting the data, did you see an overall top country that had the best diet or didn't have the highest percentage of obesity.
Yeah. I did notice a pattern which wasn't unexpected, but countries such as like Madagascar and Japan were falling on the lower side of the obesity rates, while countries such as the US, the UK, and Australia were constantly on the higher end of the spectrum and had higher obesity rates.
So that's something you were expecting to see then, that trend as well, while you were looking through it?
Yeah.
Okay, cool.
Yeah, great job.
A very interesting study to look at things differently, because there is such a bias of what people consider to be healthy. So I think that your research is really important for people to see the well-rounded side of what is actually happening. So great job.
Yeah. The one thing I want to notice is, I want to sort of tell everyone is that she obtained some of these data sets from, global databases. So, this is all of the information out there for these countries. So this is, I think one of the data sets were from the UN database. So, this is all the information that's out there for these countries. So it's, just shows like the high standard of data collection that she did.
Yeah. And a lot of data mining. So I know that was a lot of work for you. So really great job. And DJ, thank you so much for leading her along the way. So congratulations, Tiffany, on a job well done.
Thank you.
So our last presentation this evening, we have Miss Autumn, and Christina was her mentor. So if you'd like to go ahead and introduce Autumn.
Yeah, so it was my pleasure having Autumn as one of my students over the summer. She's a senior this year in California, and she's hoping to go into some animal sciences or biology. And her project really hit on a key issue in not just the US, but in a lot of places around the world, looking at plastic pollution and how it affects water quality and biodiversity. And she actually had the opportunity to take this study abroad as well, which is really, really awesome. And it was amazing to go through that journey with her and see the differences between the US and where she went abroad. So I'm really excited for her to share it with everyone. So Autumn, take it away.
Thank you, Christina. I'm Autumn Castle-Godley, and I studied the effect of plastic pollution on the water quality and biodiversity of creeks. Plastic pollution is a growing environmental concern, as I think we're all aware. It's cited as being one of 12 major threats to freshwater aquatic life, causing a decline of over 80% to freshwater populations' Living Planet Index, a measure of biodiversity, over the past 50 years. Plastic enters freshwater systems via human activity, like littering, wastewater, and drainage. And once in these freshwater systems, like creeks and rivers, it travels into the groundwater or is carried to larger bodies of water like lakes and oceans. Plastics are categorized by size, the largest being megaplastics, followed by macroplastics, mesoplastics, and worst of all, microplastics. Plastic enters freshwater systems as large pieces of litter, but breaks down into microplastics over time. Although all plastics leach toxic chemicals into the water, microplastics pose the biggest threat to freshwater aquatic life. They're frequently ingested by small organisms like fish, where they can cause cell damage and death. Over time, plastics can kill enough of these small organisms that the entire ecosystem begins to collapse. Humans are not safe from microplastics either, as they're found in most seafood and bottled water. When it comes to regulating plastic waste, the U.S. trails behind a large portion of the world. Although some states have bans targeting the use of plastic, the majority of the country does nothing to combat the dumping of plastic waste, especially into water. However, places such as the UK have many strict regulations on plastic waste, such as the London Protocol, which is a treaty that prohibits nearly all dumping of waste, including plastic waste, into water systems. The UK also has many single-use plastic bans, like on straws and cutlery. My research question was, how does plastic pollution affect the water quality and biodiversity of creeks, both in the US and Scotland? My independent variables were the region of study and the amount of plastic in and around the creek, and the dependent variables were the water quality and biodiversity. So since the US has so few regulations on plastic waste, I believe that a creek in California will have more plastic than one in Scotland. Therefore, I expect Chabot Creek in California to have a poorer water quality and a lower biodiversity than a rural creek on the Isle of Skye, Scotland. The materials I used were the API Freshwater Test Kit to measure pH, ammonia, nitrite, and nitrate, and TechoPlus LTD's digital meter to measure temperature and salinity. I began by choosing 2 creeks, Chabot Creek in Castro Valley, California, and a rural creek on the Isle of Skye, Scotland. For each creek, I chose 2 locations. Chabot had A and B, which were about a quarter mile apart, and Scotland had C and D. At each location, I marked off a section of study. I did this by taking 10 steps upstream from the initial coordinate points, and this section extended roughly 1 meter up the bank on either side of the water. Within each section, I did a plastic count, so I counted and recorded the number of visible plastics both in and around the water. I also did a species count, so I visually separated all the plants into species and recorded the number of individuals within each species. I did the same for any fish, frogs, or small insects that were in and around the water as well. I later used these numbers to calculate Simpson's Diversity Index, which is a measure of biodiversity. Lastly, I performed the water quality test using the API Freshwater Test Kit and the digital meter to measure pH, salinity, temperature, nitrate, ammonia, and nitrite. And I performed these tests every day for 5 consecutive days at each location. So as shown in Figure 1, we can see that the US has a lot more plastic than the UK does. The US has a big range from 9 to 33 different pieces, while the UK has no pieces of plastic every single day. The darker blue dot represents the mean. A t-test produced a p-value of less than 0.01, meaning the data is significant and the groups are different. Figure 2 is how the Simpson's Diversity Index, or the biodiversity, differs for each region. The US, once again, has a much wider spread of data, although we can see that the mean is slightly higher than that of the UK. A p-value of 0.18 indicates some confidence that the groups are different. Figure 3 is the effect of plastic on the water quality of each location. The US, again, has a pretty wide spread of data, while the Scotland data frequently overlaps to appear as one dot. Trend lines show strong positive correlations between variables of each graph, and linear regression tests produce p-values of less than 0.01 for salinity, temperature, pH, ammonia, and nitrate, which indicates significant relationships, and a p-value of 0.03 for nitrite indicates strong confidence in a relationship. Figure 4 is the effect of plastic on the biodiversity of each location. The data appears pretty randomly scattered, and the trend line shows little to no correlation. And a p-value of 0.72 confirms that there's no relationship between variables. Lastly, Figure 5 is the effect of water quality on the biodiversity. The data appears pretty scattered for most of the graphs, although there is a slight positive correlation for temperature and nitrite, and a p-value of 0.04 for temperature and 0.09 for nitrite indicates strong confidence in relationships, while the rest of the graphs don't show any relationship. So the UK has significantly less plastic than the US. In fact, it's a 100% decrease in plastic, which is likely due to the strict regulations. Plastic does directly affect water quality, which is shown in Figure 3, but it does not directly affect biodiversity. However, water quality, specifically temperature and nitrite, can affect biodiversity, and since plastic affects water quality and water quality affects biodiversity, plastic may have an indirect effect on biodiversity. However, it's important to note that a higher biodiversity does not necessarily mean a higher number of organisms. So biodiversity includes species richness, or the number of different species, as well as species evenness. So the individuals in each species should be a similar number for all the species within an ecosystem. And the Scotland data, although both the locations had a very, very high species richness, they had a low species evenness. So, some species had 1 individual, while others had upwards of 300, which likely produced the low diversity index for Scotland and the higher one for Chabot. Some factors that could have influenced my results are how rural or urban the creeks are, Chabot is located in a much more urban area with more human activity, as well as the size of the creek. Chabot is a little bit larger. And Scotland is located in a much harsher climate, which could affect a little bit of the water quality. I could repeat this experiment in the winter, especially the California one, because the water level of Chabot Creek raises in the winter. So a lot of the plastic that's currently on the banks would be underwater. I could also repeat it when it's polluted with different pollutants like soap or chemicals that are frequently dumped in the creek to get a better idea of the water quality's effect on biodiversity. I could expand the research by going to different locations as well with different regulations on plastic waste, maybe sort of a middle ground between the US and the UK, or I could look specifically at the different effects that different types of plastic have on biodiversity. I'd like to thank Christina for meeting with me every week and even making it work when I was across the world, just making sure that I was always like on top of what I was supposed to be doing, and I knew what my methods were going to be and making this whole thing possible. Thank you to DJ for holding the office hours to go over my statistical analysis, because that was a little bit of a struggle with all of my data sets, and thank you to Jen and my peers and everyone else at Headwaters for making this whole project possible.
Awesome job. You did great. Let me get Christina.
Great job.
Yeah. It's so interesting. So when we spoke about you going to Scotland, I knew already, just based on knowing plastic regulations, that you would see a decrease over in the UK, but that was very surprising to hear your results.
Mm-hmm.
Were you surprised with that information as well?
I was especially surprised that the UK had a lower biodiversity, because the whole time I was doing my research and performing my experiments, I was thinking of biodiversity as more the number of organisms. So when it turned out that the US had a higher one, I kind of had to go back and like relook at the definition of biodiversity and figure out, well, why are my results like this?
Kitty cat agrees.
Yeah, I'm sorry.
No, I keep chewing my way too. DJ, Christina, do you guys have any questions?
Oh yeah, I was just muted. Oh my. Yeah. So first off, I think your cat agrees that you did a great job with your project. Cat's cheering you on in the background. But my question was, which organisms were you looking for? Were you looking for macrovertebrates or what kind of species?
I was looking for any life. So Scotland only had plants. But it was a lot of plants, like constant covering. And the US had a decent amount of plants, but there was a lot of fish and insects on the water, but I was just looking for anything.
Okay.
So did you categorize them just as fish, or did you individualize them by species?
There is only one species of fish that lives in our creek. So all of the plants I did have to separate into species, but all the fish and water striders and insects, it's only one.
So you spent a lot of time outdoors. That's what you're saying this summer.
Yes, I did.
Yeah, it's a lot of fun playing in the water and seeing all the things there.
Well, I just want to say, we've already said that, you took this abroad all the way to Scotland. So also thank you to your family for putting up with me while you were on your family vacation, and asking you if you found any creeks in London, if you found any creeks in Scotland. So yeah, thanks to your family for that.
Yeah, that is true.
But I hope they had a good time tagging along also.
I think they did. They liked it.
Great.
Awesome.
Well, I just wanted to say again, thank you to our mentors for helping our students through the summer program. We do have another evening on Thursday. So it starts at 7 p.m. Eastern, 4 p.m. Pacific. And we will be having round 2. We did have quite a few students this summer, so we had to break it up into 2 nights. If you are interested in participating in our fall research program, we are still currently accepting enrollment for that. It does start on September 13th, so make sure that you enroll soon so that we can send you all the information to get you started and pair you up with one of our wonderful mentors like you guys saw this evening. If you would like to just email me directly to get more information, it is Jen with 2 N's at headwaterscienceinstitute.org. So thank you again, DJ and Christina, for all the time that you spent with our students and making this a successful program this summer. And congratulations to all of our students that presented this evening. I am so blown away by how amazing you guys did in such a short amount of time. You know, it was 10 weeks of research during their summer vacation. So well done, you guys. Everybody should be giving yourself a round of applause. So I hope to see everybody on Thursday for us to have our round 2. So have a good night and let us know if you have any questions.